such as that, mostly with biological responses in response to micro nanoplastics in both in vitro and in vivo systems. But today I'm going to present some project that I've also been part of. That is a field sampling campaign in this plastic bottle recycling facility, still related to micro nanoplastics. Yeah, so this was all part of an EU-funded project called Plastics Fate, which was part of this CUSP collaboration of several EU-funded projects that all focused on human health and micro nanoplastics. It ended in April this year. If you want to read more about this research, you can go access this link. And this figure, it sort of demonstrates why plastics has become literally a growing cause of concern. Around this time point, we have exceeded 400 million tons of plastic being produced each year. And it's expected to increase and even surpass 1,000 million. So it is an issue that we have to deal with, definitely. And as you can also see, most of it is plastic is discarded, being used in landfills and such. Some of it is burned, and just a small portion of it is recycled. If you focus on EU, it's a bit different. We produce much less. Most of it is produced in Asia, of course, and we recycle quite a bit more. But still there is room for improvement. And if you look at the different plastic polymers, polyethylene terephthalate, for instance, to the left there is one of the plastic polymers that are being recycled the most. And in Norway, we are very efficient, and we actually recycle most of this polymer. Norway and UK is not part of the European market here, but we share this. So back to micro nanoplastics. The definition is microplastics are having a diameter between 1 micrometer and 5 millimeters. Nanoplastics, if you follow the definition that we usually use in the field, it's less than 1 micrometer or 1,000 nanometer in size. Different sources, primary, some are being produced for specific purposes, for research purposes, for cosmetics and such. And you also have secondary sources of MNPs, which comes from degradation of larger plastics items. And this is, usually happens over longer time periods, but as for instance in a recycle facility, this can also happen very, very fast. Exposure routes, you have ingestion and dermal contact, but from an occupational point of view, it's of course inhalation that we're mostly focus on. And then these figures are summarized the main findings so far in the field, where you see studies saying that they see translocation and inflammatory responses, DNA damage, and even some reporting cytotoxic effects. And then the question is, are nanoplastics more harmful than microplastics? They can potentially cross biological barriers. I want to say potentially because the research is still going on. And for inhaled plastics, they could maybe then translocate from the lung epithelium into the bloodstream. And the high surface area volume makes them more reactive. And they may act as efficient carriers of harmful chemicals and microbial contaminants. And if you look at the figure on the right side, you see that the smallest particles, bigger portion of these, can deposit in the alveolar region of the lungs. Tissues and organs where plastics has been found. I'm sure many of you have read about this in regular newspapers, especially this paper to the right there where they claim to have found plastics in the brain. This is also a bit up for discussion. But it's clearly that more and more studies are coming claiming that they see plastics in various tissues and organs. So MNPs in workplaces. So kind of as I mentioned already, micro nanoplastics can interact with the biological system and represent a possible source of health risk in exposed workers. The presence and sources of micro nanoplastics in workplaces have received little attention. I think most of what we know about it comes from the flocking industry. Plastic waste management and recycling is the sector where workers may be exposed to microplastics and nanoplastics in the air. And airborne nanoplastics, particularly the nanoplastics, have been poorly characterized, mainly due to their small size and limited detection of current analytical techniques. So the aim of this the study was to characterize airborne micro nanoplastics in this recycling facility by assessing particle sizes and concentrations, as well as polymer mass concentrations. So this is just an overview of what's happening in this factory we visited. The example here is for a high-density polyethylene plastic, but it's the same principle for a PET plastic as well, which is used for these plastic bottles. So the whole process is you collect it, you fragment the plastics, you heat treat it, and then you sort it. And then in the end, it's being thermally degraded and formed into these small plastic granules, which is then sold to other companies that can use it for producing new plastic bottles. And it's especially then at the shredding stage of the larger bottles, and also where you thermally degrade the smaller fragments, where you potentially could have airborne micro nanoplastics arising. So this is kind of the setup for what we did. So as I said, at this site, the polyethylene terephthalate bottles are recycled into the granules. You can see it on the bottom there. Shredded into flakes, cleaned and heat treated. And we placed then stationary instruments inside the factory area and also in the office area to use as a background. And there's a lot of potential sources for the micro nanoplastics in here. You have the heat treatment of the plastic, silos with shredded materials, conveyor belts, and trucks operating inside. So a lot of dust can be generated and come from these different sources. The measurement and sampling equipment, scanning mobility particle sizers, which counts the smallest particles in the nano-size range. And then it's sort of the APS, the aerodynamic particle sizer, complements this instrument by measuring a bit larger particles. And then it's the two at the bottom, they collect samples, stationary air sampler, or it can also be used for personal air sampling. Collects the inhalable fraction up to 100 micrometer. And then the SUTAS cascade impactor, which sort of collects different fractions, different size fractions, particles smaller than 0.25 micrometer in size, and then up to 10 micrometer. And if you see the downstream analysis, it's this one we used for the pyrolysis GCMS to analyze the plastic polymer type in the different fractions. So the main findings: airborne dust inside the recycling hall was very low, 0.21 milligram per cubic square meter. This is well below the occupational exposure limit for organic dust. We don't have an exposure limit for plastic at the moment. Higher levels of airborne nanoparticles. If you look at the results for the graph from the SMPS, you can clearly see a lot of nano-sized particles. This was in the blue one is inside the factory, whereas the background in the office area is the yellow line. The same you see from the APS. And pyrolysis GCMS, I don't have the numbers, it's still being analyzed, but by looking at the raw data, we can see that we have PET plastic in the range of 0.08 to 43 microgram per cubic cubic meter. And we have some scanning electron micrographs of what we believe to be plastics in this nano-sized range, and also dust that was collected inside the inside the factory area, also looking like plastics. So the simple conclusion, our findings indicate that workers may be exposed to airborne micronanoplastics emitted during recycling of plastics. With that, I would like to thank especially my colleagues, Øyvind and Paul at the chemistry department, who did the sampling campaign, and they also performed it in other factories. And also our collaborators at Aachen University who do the pyrolysis G6. And this is a picture of the toxicology group at STAMI, where we went to Sandefjord to hear about the whaling industry going on there. It's quite a long time ago. Yeah, thank you.